morning, everybody, and welcome to Grand Rounds. Next week, our speaker is going to be, I'm going to show my ignorance here, is Luigi Ferrucci from the NIH, and he'll be talking about rethinking the metrics of aging, the example of sarcopenia. Who knows what sarcopenia is? What is it? Change in body temperature? Oh, curvature of the spine. I see. So it's a orthopedic musculoskeletal infirmity. Okay. Well, I assume it's related to what our division of aging and geriatrics does. So Patrick Brown is the host. I assume Brett and Dev and everyone had a hand in inviting him, so that should be interesting, or at least those of us who are aging. Today, I want to make one announcement before we start. Jürgen Kaiser and Craig Tenke received a R21 from the NIMH for a project titled MDD Treatment Response, Major Depression Treatment Response and Functional Connectivity of Brain Regions via Reference-Free EEG. So it's a study based on the EMBARC, which is a biomarker development program from the NIH. So congratulations to them. It's really a pleasure to have today one of our own faculty speaking, Dr. William Fifer. So Bill was educated in terms of his bachelor's degree at Georgetown and then received his MA and PhD in psychology from the University of North Carolina at Greensboro. He then migrated north and was a research fellow in developmental psychobiology at Albert Einstein, which is presumed where he connected with Myron. And when Myron moved here, brought Bill. Is my inference correct? And the rest is history. Bill started as an assistant professor and rose to become professor of psychiatry and pediatrics, associate director of the Sackler Institute for developmental psychobiology, and also holds the title of extraordinary professor in obstetrics and gynecology at the University of Stellenbosch. Bill has been really a pioneer in the area of developmental biology in the area of clinical studies of that, and that's what he's going to speak about today, but in doing so has pursued a sustained line of research in studying sort of infant brain and behavioral development, and in that process has received funding from a variety of sources, incurring most recently from NICHD, from NIMH, and also from the Bill and Melinda Gates Foundation. I'd like to know how he got in there for some of that money. But he's had collaborations with a lot of people in our faculty and has really, I think, been a true example of the value and the capacity for collaborative research. So it's a pleasure to have him speaking today on his work and, in this case, the earliest markers and mediators of neurobehavioral development. So please welcome Dr. Bill Fifer.
Thank you all, and thank you for showing up. You know, you mentioned that, Jeff. I kind of wear that badge of honor of collaborative research, and in fact, some of the people that are mentioned up here, I only listed those that I'm going to be discussing the work that they were involved in today, but I've been so blessed and grateful by having many, many people in this room that I've been able to collaborate with. So I'll try to highlight that as the time goes on, and I just feel compelled to also say about that extraordinary professor thing. It's from Stellenbosch University, but you should know that if you're a professor at Stellenbosch and you teach on campus, you are the ordinary professor. If you're off campus, you're the extraordinary. So to be fair, although I feel extraordinarily, again, blessed to be down there working with those folks. So I'm going to tell you, I'm going to be a little bit, I don't want this to be a personal journey or a lab journey, but there'll be a smidgen of a travelogue in there. But if I gave you that personal history. It'll probably take about, the interesting bits would take about 2 minutes, and the rest of it would take way too long. But what I am going to do is highlight a few of the recent studies that we've been involved with that really, I think, speak to what our overall mission is to understand a little bit more about what that newborn infant brings with him or herself into the world, and what we can tell early on about what might be the potential trajectories for that individual. So I will first, you will have seen 2 of these, at least those of you who have heard me speak before. There'll be 3 slides I'm going to repeat. This is one I have to show all the time because it is the beginning of where we started. In fact, I look at this picture, it must have been taken with a Brownie camera or a Polaroid or something, because it is so old. And what it is it's a picture from my dissertation about 100 years ago, where we were able to ask a question of a newborn about, where we began a series of questions about what that newborn knew about his fetal world and what preferences that fetal experience brought with him or her to the newborn period. In this case, we were finding out that newborns, in fact, had a preference for their own mother's voice on the first day of birth. And in fact, we went on to ask a number of other questions about their capabilities for remembering something about their language, not the words, but the cadence, the prosody of that language, that in some way played into the early attachment to the primary caretaker. But that particular study, which I don't even want to think about how long it took, we needed babies to be wide awake and alert. In fact, that study probably took about 2 years to get a grand total of 20 subjects, just to be able to have the right subjects at the right time, get the right structure to be able to work. And so, more often than not, this is how you find your subjects when you're walking into a newborn nursery. Similar to a few looks I'm getting right now, well, not yet, but there also are, this is even more likely, where the babies aren't quite ready to cooperate. No matter how interesting your experimental question might be to them, they don't seem to think it's going to go that way. And sometimes there are those that are even less interested, and that's a harder group to deal with. But fortunately for us, this is where the lamppost is shining. Babies are sleeping most of the time, and their brains are very, very active. And in fact, that's where we've been able to ask most of the questions that I'm going to be telling you about today, is while babies are sleeping. So this is to help you plan your either your nap time or when you have to look at your emails or texts or whatever. This is what I'm going to try to accomplish today. And we've been working on many of these aspects of what we think is helping us to build an early profile of, again, of that infant brain and begin to try to understand what brains are going to need a little bit more intervention than others. So I'm going to be talking about our measure of fetal brain development, autonomic nervous system function during sleep. Well, I'm going to be talking about the same thing for newborns and young infants. I'm going to skip this, although I'll be happy to talk about it later, where we're working on a lot of EEG responses, particularly with our colleagues in pediatrics. I'm going to be talking about what resting state EEG activity can tell you about the capabilities that newborn is bringing to the ex utero world. I'm going to be talking about learning capacity. We won't be talking about this today, although it's one of my favorites, especially being able to measure sleep as the mediator of so many things that we know are important. And I'm going to then talk, as we've gone into the geriatrics realm, where we've gone out to toddlers, out to 15 months and even up to 2 years of age. So that'll be where we'll be heading. I'm going to give you a little primer, very brief primer on markers of how well the autonomic nervous system is doing its job. One of them is a very simple one, which we have to take care of every single day of our lives, but if infants and the rest of us have to do the same thing during sleep, which is, while you are breathing, your heart rate goes up and down during each breath. That's called respiratory sinus arrhythmia. It's a marker of vagal tone, or the parasympathetic arm of the autonomic nervous system. It is something easily measurable. In fact, you could see right here, you just have to look at the differences in heart rate with each breath, and it gives you a really good clue as this interaction develops of how the autonomic nervous system is developing and is allowing us to begin to get at trajectories. Now, this is during quiet sleep, where the baby's sleeping like a metronome, and it's really a nice window into how the system works. But much of the time, Babies are in another sleep, and another sleep state. This happens to be REM sleep, where the respiration is not nearly as regular. And there are periods of apnea, which are very normal in a newborn infant. And then you'll have things that are these augmented breaths, otherwise known as sighs, which have a very different interaction with heart rate. But again, this interaction about how the autonomic nervous system reacts to and controls the integration of respiration, heart rate, blood pressure, temperature, and in this case, movement, where you get a big movement and a big change in heart rate, are nice ways to go in and probe, again, if the system is working properly. When you do that, you can find that you have to start paying attention to a lot of things in order to understand how this is developing over time. One of them is even across different 2-week intervals. These are preterm babies starting at 32 weeks. When you want to get a nice measure, this is one of several markers of heart rate variability, a marker of autonomic function. You can see you have to pay attention to sleep state. This is a baby who's in REM or active sleep, a baby in quiet sleep. If you want to go in and give a number, a marker of how well that system is maturing, you have to pay attention to that. You have to know a lot about their actual age. Even over these first 4 weeks of development, you can see there's a marked change in this marker of autonomic nervous system development. And then you can begin to follow children up and see that, again, it's like a growth chart, but it's a growth chart of the autonomic nervous system, again, what we think is a viable and feasible practical marker of brain development.
How are you deriving heart rate vulnerability from those parameters?
Yeah, it's really, it's heart rate variability. And what it is it's again portraying all the different rhythms, not just the one I mentioned with respiration, but there are many other coupled respiratory and cardiac functions that are showing us that the brainstem, that the autonomic nervous system is doing its job. The vulnerability part I'll show later when we're showing how.
How are you deriving that parameter?
This is a parameter where you just take your beat-to-beat intervals, your RR intervals, and you devise different ways to look at those changes from one beat to the next, not just from neighboring beats, but over the course of minutes, hours, and days. Here's another metric of those of heart rate variability taken from one of our large samples that Michael Myers, who, by the way, I'll probably say his name about 23 times, and if I don't, it was a mistake, because Michael is behind almost all of the work that I'm going to be talking about today. This is when we're looking at this heart rate variability measurement during this transition to what we know is the beginning of the vulnerable age for sudden infant death. A lot of our research, I won't be talking about it today, but has been focused on vulnerabilities and mechanisms underlying sudden infant death. But here is a portrayal of, here's heart rate variability. We'll just look at quiet sleep right now since this is a busy slide. But a newborn baby within the first couple of days of life, this is the average of this particular measure of heart rate variability during quiet sleep. And then when they're a month older, when they're going into the vulnerable period of SIDS, there are marked changes in how it is that the autonomic nervous system is responding to, is orchestrating these interactions between heart rate and respiration. And we make sure we pay attention to both sleep states. We also have other measures. This is a global measure of heart rate variability. Again, this is that beat-to-beat measure of heart rate variability we think are very, very important, not just in gauging development, but also gauging vulnerability for sudden infant death. Here's an example of where we can take that same metric and look at a newborn baby, but notice differences in groups of babies whose mothers did not acknowledge using alcohol during pregnancy versus babies whose mothers said yes, they had at least one episode, in this case of binge drinking, where on average these babies' portrayal, this quantification of heart rate variability is significantly less in the newborn period on day 1 of life and day 2 of life and day 3 of life than if they weren't exposed to alcohol. This is just by way of showing if we didn't pay attention to active sleep by itself and looked at all sleep states or put this quiet sleep in the calculation, we wouldn't have seen a difference. So some of these vulnerabilities, if you will, these deficits in autonomic function, come on down here. I'm so surprised there's so many people. When Eric had even more people in this, he said, come sit at my feet. I won't say that, but come on down. And come on. Unless you're trying to get out early, in which case stay there. But there are plenty of seats. So And then I just want to show there's always more things that you have to pay attention to. And one of them is that if you're going to have this quantification of autonomic function, you not only have to pay attention to age and to sleep state and actually time after feeding and body position, you really have to know something about sleep state itself. So here's an example where you go back and you can look at heart rate variability in the fetuses. So we can start this journey of looking for this developmental marker even during the fetal period. And here's just showing early pregnancy, 20 to 24, 28 to 32, 34 to 38. You see this nice increase in an autonomic capability or autonomic complexity, if you want to call it. But once you get out here to the later period of 34 to 38 weeks, you now have to start paying attention to fetal sleep states, because fetal sleep states 1F is the equivalent of quiet sleep, this is the equivalent of REM sleep, and this is the equivalent of being an active, awake fetus. We can figure that out based on ultrasound. So you can see, you can begin to get these markers, these trajectories, but you have to make sure that you're measuring your fetuses, in this case, and babies under the right circumstances and in the right context. And then you can begin to look at things like these numbers of babies from the Safe Passage study I'll tell you about in a moment, where at this juncture it was over 8,000 babies had been studied. But you can begin to see that you get a nice portrayal again of this growth function. Again, this is the average growth function, and what we're really going to end up being interested in are different trajectories. Not all fetuses will respond the same way. So the fetuses who start low, go higher, and then return or stay at baseline are the same steady state. Those are fetuses we're going to have to pay attention to, we think, based on their early profile. And there's another wrinkle, of course. You know, you're trying to figure out all these different things about, wait a minute, I want to be able to assess this infant relative to his or her peers. And you have to pay attention to sex as well. So in fact, these measures of, in this case, even basal heart rate, there are no differences really early on in pregnancy, but by the time you get to the last few weeks of pregnancy, there are significant differences in female heart rate versus male heart rate, and in short-term variability. Again, one of our markers of autonomic function, something we hadn't anticipated, but in fact is there starting quite early on. So as we almost finish this primer, part of why we got so interested in this is because, and now it's pretty well recognized that sudden infant death is caused by dysfunction in the brainstem area, probably due to some prenatal exposure, some insult, some hypoxic incident that happened at the wrong time and at the wrong place during this early part of development. And so we, Hannah Kinney particularly, I should have had that reference down here, has spent a lot of time looking at serotonergic activity based on her autopsy work on babies and fetuses who didn't survive in these particular areas, where serotonin is a big player. Now, many of you in the room are realizing that this serotonergic system is very, very important in cognition and affective behavior. For us, we've been focusing on the lower brainstem and this orchestration of respiration, autonomic control, chemosensitivity, which we think is terribly important in SIDS. So though we've been focusing on this and those branches, I just want to remind folks that early insults that have affected this part of the brain and these circuits are probably also going to emerge and be reflected in later cognitive and affective problems. Okay, so I'm going to give you a few slides on, well, how do we measure some other aspect of early brain development? This one happens to be using very non-invasive, high-density EEG. This net goes on in under 2 minutes. Most of our babies don't even arouse when we put this net on. And it allows us very quickly to be able to gauge a very direct marker of brain activity and connectivity. Come on down. And so we are blessed that we have this particular tool. And in fact, one of the people who's just walking in here, Phil Grieve from Pediatrics, is one of the people who's helped us make sure that we can exploit this tool to its fullest. And we can begin to take this signal, which you gather at the top of the brain, and you get this noisy little wave, but then you can break it down using Fourier analysis into its component waves. Just as you would with light, you can break it down into the waves that we all now have recognized, oscillations in the brain, beta, alpha, theta, delta, gamma, and others, which we think are reflective of very important brain processes. And so we can do this while babies are sound asleep on the day of birth. When we do that, we can start seeing differences that are emerging already, just as a function of a few weeks difference in gestational age at birth. These are babies born at 36 weeks. With their EEG activity, this particular one happens to be high-frequency activity. When they're in REM sleep versus quiet sleep, you can see this changing just from 36 to 40 to 44 weeks, and also changing by sleep state. So again, this marker also mandates that you pay particular attention to not just sleep state, but the age of that baby. You can begin to ask many different questions then about effects of prenatal exposure on different brain areas using this technique. This particular study was done in the neonatal intensive care out in Rapid City, South Dakota, part of our, I'll tell you in a bit later, about our Safe Passage study. What this busy graph is showing, but I think very carefully is showing that wherever you see a dot, a colored dot here, it means there was reduced power in these parts of the brain. Here's the nose, here's the ears. There was reduced power at different frequencies in babies who in the intensive care unit whose mothers say they drank during pregnancy but didn't smoke, those who said they smoked during pregnancy but didn't drink, and those that did both, where we see very, very different, much lower power, in particular in these parts of the brain regions, based on these exposures in utero that we can recognize now within days after when the baby was born. Yeah. Yeah.
One of the problems with electrical recording or stimulation of the brain is the impedance of the skull and the effect that that has.
Mm-hmm.
Given that infants have less well-developed and thinner skulls, is the fidelity of EEG in infant neurophysiology more accurate?
Yeah, no, it's a good question. So again, it's can we trust the fidelity, or how do we feel about the fidelity of our signals with these young babies' brains coming from skulls that are very, very thin? In fact, it's an advantage because, in fact, there's less of this sort of smearing across the scalp when you're recording your EEG, and the impedances can be quite high, but because of these amplifiers, these particular amplifiers, we don't have to deal with that question. And in fact, we don't have to deal, you have to be careful, but you don't have to deal with environmental noise all that much either. So it's a very effective technique. And in fact, I don't know if Dev is here, but we're going to be using this same device with some of his patients looking for early brain markers, connectivity markers at patients at early risk for Alzheimer's. And again, this very high impedance, but very valid measure will work there as well. I want to bring this one up, even though you could hear more about this if you want to go to Pediatric Grand Rounds on Friday. But there's been a wonderful study going on with the Nurture Science program with Martha Welch, Michael Myers, Myron Hofer, and others in this group, Phil Grieve, where they have gone in and engaged in a very interesting and very powerful intervention with mother and infant during their stay in the NICU, where their really focus is on early emotional regulation, early parent-infant interaction. And they, by measuring the brain activity before and after their interventions, and also comparing it to infants who have standard care versus infants that have infants who have had this family nurture intervention, are able to find very clear-cut differences early on in life for those. This with these colored dots means there was an increase in power with the infants who had the intervention versus those that don't. So they already have many, many behavioral measures, but here's a brain measure showing truly that, in fact, the brain was affected by this intervention within just weeks, weeks of the intervention. Again, we can also use this same tool in other ways. We can look at resting state, which is interesting, but you can also push the brain a little bit too. In this case, Joe Eisler did a study where he gave two different tones to these sleeping infants. Down here is portrayed a standard tone that was given most of the time, but every once in a while in this thing called the oddball or mismatched negativity experiment, you put in a deviant tone. And then you start looking at how the brain is doing something different when it recognizes, first, it's just a tone. That's what this brain average response is reflecting right here. But then, milliseconds later, the brain is recognizing sound asleep, that this thing is different from the one I've been hearing all along. And you can start doing these frequency by time plots to show there was a big increase in this theta or alpha power here at about 300 milliseconds into that stimulus, where the brain appears doing something else. You're getting marked changes in beta activity and gamma activity at different times when the baby is listening to this particular sound, sound asleep, but all of a sudden is noticing, that's a little bit different than what I just heard a little while ago. So, and this is a metric that not only can you then measure or quantify the different power ranges, but you can also go in and find out exactly where those differences are occurring. What part of the brain that doesn't necessarily have to do with hearing, but something that's noticing a change in environmental stimulation. We can go in, find it, and measure it. We can do one more thing, and then I'll get out of the techniques and maybe into some of the cohort data. We can then ask the brain about its ability to be able to put 2 and 2 together and end up with the right number 4 in this case. And what we did, it was have a classical conditioning paradigm where we have babies, again, sound asleep. There's these speakers right near their ears. We have the net going over their head, and this little tube is able to present a gentle puff of air to the eyelid at different times during our experimental protocol. Again, sound asleep baby. This is very similar to, in fact, it's often called eye blink conditioning. But what we're doing is not looking at blinks as the baby's eyes are already closed. They're just closed, and they get a little tighter when that gentle puff of air comes to their eyes. So what we do is pair a tone for 100 milliseconds. At the end of it, we give that puff of air. We do this about 200 times. And every once in a while, we put in probe trials, like every 10th trial. We don't do the tone. I don't do the puff of air. We just do the tone by itself. And what we do is we measure what is that baby going to do now after the tone and puff have been paired about 100 times, when all that baby now is doing is hearing the tone by itself. And what you can see is, this is taking 25 minutes during this session in the newborn nursery. During the first 5 minutes, there is no difference in the amount of responding to the tone by itself, but gradually over time, babies start blinking to the tone alone that is predicting the puff that's going to occur. Again, the vast majority, around 90% of our babies would learn this while they were sound asleep. We can take that measure and also go in and measure something about what's going on in the brain at the same time. So we can go in, and in this case, the first half of the study is portrayed by the dark lines, the second half by the dotted lines. We could see, what was the brain doing when it was listening to those tones in the first half of the study? And then when we think the babies are starting to or have learned the association, what's the brain learning during the second half of the study? And by about 500 milliseconds into the tone, you start seeing this difference in brain activity. Again, in this part of the brain, and this happens to be high-frequency activity, where we can see that the baby's brain is reacting very, very differently to this thing that just meant a tone before, but now has come to mean something else while they're sound asleep. Now, again, this is an important thing for babies to learn, and I hope they forgot it. I don't want to be out there on the subway when these kids are 15 years old and my cell phone goes out and they start blinking like crazy. Hopefully that'll be gone. What these kids are able to do, though, is show that there's an ability for the brain to put things together, to make associations, again, even while sound asleep, which is important for all sorts of reasons, which we can get into in a bit. We can look for the auditory evoked potential and measure that and make sure that, yes, indeed, that the auditory system is working. But then we can go in and look and focus in on that time, that difference of what's going on in the brain, when and where it's happening when the babies have learned this. So here's the cohorts I'd like to tell you about. We are fortunate enough in being involved in a number of different cohorts. I'll hone in on 3 that we're engaged in at the moment. One of them is one on late preterm infants. This was largely led by Nina Burchin, who was with us a couple of years back, where, well, I'll tell you why, and then I'm going to tell you about the other ones in a moment, where it's been found that these infants who are born just in the neighborhood of 4 or 5 weeks early are very different than their full-term counterparts. You can see that visually. But they have been treated as full terms forever. In fact, these babies born at 35 and 36 weeks go right to the standard newborn, healthy newborn nursery. If you're born at 34 now, you used to be considered full term. You get that little checkbox, well, I made it to 34 weeks in pregnancy, I can have my baby at any time. Well, obviously that isn't true. These babies aren't quite ready for prime time. They haven't been in the oven as long as they maybe should have. And it's reflected not just in their size, but in other, we think, meaningful dimensions about how it is that they're going to have to deal with this ex utero planet that they've emerged onto without maybe being properly prepared for that. So they are at medical risk, 3 to 7 times increased morbidity. There are various times when many of them have to be rehospitalized, usually having to do with respiratory problems or jaundice. But if you look at them at different ages during the pre-K period, now retrospectively, it's been found to be much higher risk of some delay in pre-K, on average, about 10% lower scores in some of the basic standardized tests are given in the first grade and third grade, 30% higher odds of needing special education. Much older, they are at 70% increased risk for various hyperkinetic disorders. And later into adulthood, there seems to be an increased risk for emerging cerebral palsy type symptoms based on the age at which these babies were born. This is just kind of a nice little metric of the prevalence of special education needed by when you were born. So if you're born, we knew this, that if you were born very premature, 24, 25, 26, you are going to be at high risk of have a major need for having special education help later. Here's the group of late preterms. They still are at a lower need than their much micro-preemie counterparts, but they still are at a greater need than these babies born at 37, 38, 39. And there really is a dose response once you have the numbers to be able to show these sorts of things. In fact, we had the numbers here, thanks to Howard Andrews, I don't know if he's here today, where he got two entities, the education department and the health department, to play in the sandbox together. And we got information about gestational age and other birth information, and then what the scores were on standardized tests in the second grade when these kids were asked to take these tests. Whoops, sorry. There we go. And what we're able to see is that as a function of gestational age, your score on this reading task was significantly different by each week, specifically right here. And even though the differences in the scores, like 50 versus 51, that isn't big, but what it does is it shifts the distribution. So now these kids down here are 33% increased risk of having a severe reading impairment. So it changed kids down near the bottom of the distribution. Same thing, as it turned out, was true for math. But again, we don't have the same sort of early interventions having to do with math, which maybe we should, but the reading ones happen to be noticed earlier. And so this happened to be also true for math scores. And this probably comes as no surprise. These people are coming into the world with very different brains. In fact, here's a slide from Hannah Kinney, one of our colleagues, collaborators on the Sudden Infant Death Project. This is based on autopsies, but you can clearly see not just even the gross picture of what the brains are looking like. This is a late preterm baby's brain. You can see this difference, but you can also see the differences in cortical complexity here between 35 and 40 weeks. I think I got this slide from Holly Moore, who is my source for all things brain, so I thank her for that. She's also the guardian angel in our new facilities, which you're welcome to come by the way, the new Sackler Institute facilities on the 4th floor. If you get a chance, come on by. Anyway, this is what's different about those brains, and you can begin to measure that. So you can start looking at all the different parts, of the regions of the brain, frontal, temporal, parietal, occipital, and different frequency bands. In this case, I'm going to look at low alpha just as one example, where you can start seeing that the activity is very different, whether you're late preterm, early term, or term. Again, average 35 weeks, 37, or 39 to 41 weeks. You can see this change, and some change more rapidly than others, and some have a steeper slope, but we can now begin to measure these things as a function of, again, whether you were born just 4 weeks early or 2 weeks early or on time, and have a starting spot, a starting place. And so then we also can look at that other marker we're interested in, which is heart rate variability. In this case, that respiratory sinus arrhythmia by gestational age at birth. Here are the differences in active sleep. Again, just 2 weeks difference from here to here to here, and you get differences based on what sleep state you're in, which again, we are telling us a lot about how ready is that baby to be able to interact with the outside world in very, very meaningful ways, because our autonomic nervous system clearly is involved in not just, which is very important, keeping us alive, but things like attention and emotional regulation. That balance, if it's disturbed, is going to disrupt all of those functions. Then we can get out to when they are really closing in on geriatrics, where we can bring these babies back at 9, 15, 18, and now out at this age. And then we can start looking at associations between what they did as a newborn and these various phenotypes, from the neurocognitive development ones to tests we have for imitation and memory and speech discrimination and emotional regulation. That's what this BITSE is, the Inventory of Toddler Social Emotional Assessment. I think that's what the A stands for. And we can begin to look at, were those early markers? Are they telling us anything about where this baby is going to end up? In this case, we're using that inventory of social emotional regulation, the BITSE, and we're looking in different frequency bands. This is beta, this is alpha, this is low gamma, all important bands we want to look at. And if you had a low power in your beta frequency band, this is this group right here, the higher the number here, the more problems you had, the higher you were for risk for autism based on the BITSE autism risk scale. So again, low power, higher risk, medium power, lower risk, and high power, what we think to be normal, is at a lower risk. And again, this was reflected in different frequency bands in different parts of the brain. So already at birth, we're beginning to see these reflections of what can be happening, and this was done at 2 to 4 months of age. This is, again, to portray something about the individuals and not just the group effects that we're looking at. Here, again, is the BITSE problem score. Again, the higher the score, the more problems you have in this domain of social emotional regulation. This is looking at comparison with our other metric of brain development, heart rate variability. So if you had low heart rate variability as a newborn, you were going to have more problems on this BITSE score at 2 to 4 years of age. And if you had high heart rate variability as a newborn, you were less likely to have any problems show up on this particular scale. So there's another cohort we're working on, which when I agreed with Jonathan Posner about giving this grand rounds, I said, oh, I'm going to have this great story to tell about the Safe Passage study because it's going to be all done and we're going to have all the work out there ready and finished to tell you a wonderful story. Well, in fact, that study ended 180 days ago. I know that exactly because I got that reminder today of you are not in compliance, your progress report is not in yet. And that happens after 180 days after the grant is over. So we still don't have all of the data analyzed. We have some data that now is under review that I wish I could tell you about, and I can't since we're on camera because it is under review and we've been asked not to speak about it, but I can tell you that we're finding very meaningful relationships between exposures prenatally and vulnerability to stillbirth and sudden infant death.
Yeah, we can turn off the camera.
Oh, we could? Yeah, well, I'll still probably tell you individually or whoever's interested, but we could do that, couldn't we? In fact, maybe when the question and answer, we might do that. Anyway, this wonderful study funded by NIH, a few different agencies at NIH, we were able to study 12,000 maternal fetal dyads, 7,000 in South Africa, where there's a very high incidence of unexplained stillbirth, sudden infant death, and fetal alcohol-related disorders, and in the Northern Plains, North and South Dakota, where half of the participants were Native Americans, half non, where again there is a higher than normal incidence of exposure to these things in utero. And we were able to get a lot of epidemiological information. They did such a wonderful job. They know not just when the mothers drank and how much they drank, they know how much ice was in the drink and did they share the drink. Really wonderful metrics of exposure, and not just of drinking and smoking and other drugs, but nutrition, psychosocial measures, et cetera. There's genetic analyses ongoing in all of these fetuses, infants, and their parents. Michael Myers and I were the head of the physiological assessment unit, where we measured all these things in all of these babies, both during pregnancy and when they were born. And then there were many placental analyses done because, again, if you want to have a good measure of how things went during pregnancy, just see if you can carefully read the placenta, which is a wonderful diary of how things went during pregnancy, and more attention is being paid to that. And then, unfortunately, but we knew it was going to happen as part of this study, there were many babies and fetuses who met their demise during the time of our study. And so Hannah Kinney and her group were looking at many of the selected neurotransmitter systems and circuits, particularly serotonin was big among them, of the infants who went on to die. Following that study now, though, is the ECHO study, Environmental Influences on Child Health Outcome. Many of you probably know about this already. There are a few of us here at Columbia who are part of this ECHO initiative, which is going to be 50,000 babies across 83 different existing cohorts of mothers and children who NIH, because the National Children's Study wasn't going to work, it wasn't going to work. It was going to end up costing billions and billions of dollars. So instead, what they've chosen to do is take existing cohorts. So there are cohorts out there interested in early markers, early vulnerabilities for asthma, for obesity, for neurocognitive growth, for development, for growth, for health and well-being. Got all these people to play together in the sandbox and start following them longitudinally and harmonize existing measures and standardize new measures so that with our cohort, will now be measuring things having to do with asthma, which we never have done before, and obesity. And so hopefully by the end of this is to have 50,000 children, which will become a national resource to use by ECHO investigators. And the other very big difference here is within a year of when you've collected your data, this data is gone out to everybody. So you have a year to make sense of it. And again, this isn't by the end of the study. It's every year you have to put in your data you've collected by that year and then start working on it and then send it out and let everybody use it. So it's a very different model. But I think it's one that I think will justify some of the costs that are going into this, and one that I think can be used by a lot of researchers in a lot of different fields, because again, these kids are going to be followed until they're 20 years of age. So the other cohort that I'm interested in, and Jeff mentioned Gates funding, and I was fortunate we got was called a Grand Challenge, a Phase 1 award, to go in and take a subset of the babies we studied down in South Africa to begin to look for early trajectories in fetal heart rate variability that then might be a prenatal marker of perinatal brain development or early brain development. And so we did, on many of these kids, we were able to do EEG when they were born. This thing had to have a name, so I called it BEATS, which it's a good thing I'm just calling it BEATS, but it stands for Bioelectric Evaluation of Antenatal Trajectories. I'm not going to say that every day. But the gist is by looking at heart rate variability in very exquisite detail, which I'll show you how we do that, we can get at markers that people could never get at before and how fetal brain is developing. And this logo is telling us that what we can look at for rhythms in the heart can tell us so much about that developing brain and the feedback from the brain back to controlling autonomic output for that fetus to be able to survive in utero and ex utero. I also show this because my nephew drew this logo, so I had to show it. I told him I would every time I get a chance, so I just did it. It brings me to nice places too, by the way, speaking of nephews and children, places I've been able to bring my kids, places where Michael Myers and several of our team have visited along the way. One of them is in the Badlands of the Dakotas. This happens to be in South Dakota. We went to nearby Indian reservations, and we went to two urban sites, Sioux Falls and Rapid City in South Dakota, and Spirit Lake in North Dakota, where again, we were welcomed by the people there because they did know that many of their infants were dying. Many of them seemed to be resilient. Some of them clearly would have needed extra care from the get-go if we only knew maybe who to target. So they were very invested in our being there, and still are, because these kids are still now going to be followed up. This is the other site which we went to, Cape Town, and you can see where it wouldn't be a bad place as you fly into to get this view of Table Mountain. The hospital we work in is just over here. And it's brought us to places like this other part of South Africa, Limpopo, where I just remember so vividly. I was looking at this image this morning, just standing there. I mean, this is the cradle of civilization, and I'm not the spiritual type, but I was moved by just standing there and realizing that it was much the same picture. We're talking thousands and thousands and thousands of years ago, and we were blessed to be able to be there and to work with the people off in a nearby area looking at other interventions in early childhood. So how do we get these new measures where we have some confidence? Heart rate variability has been around forever. You know, you go and have an ultrasound and you hear the heartbeat, you hear the whoosh whoosh coming through on the ultrasound. But that particular measure of heart rate is averaged over seconds, actually. You're getting these averaged heart rates as the ultrasound is bouncing off of the heart. What we're able to do with this device is get at the differences between beats, back to Jeff's questions earlier, the difference between heartbeats, interbeat interval in the millisecond range, which allows us to then go in and look at these before we were unable to look at these very fast rhythms in heart rate, which were telling us about autonomic function. Now you get this signal on the mother's abdomen, but online, the software removes all of the mother's QRS, all this other muscular noise, and what you're left with is the fetal QRS intervals. So you can get a fetal heart rate to the near millisecond with millisecond accuracy. And you can do this non-invasively, and you can do this not just in the clinic, but you can also do it at home. So our goals in this early cohort was to measure autonomic function with the Gates Challenge money. We now just this week, got phase 2 Gates money, which is considerably more money to be able to do more of following up these kids. And we're grateful for them to do this because they care about this particular device can be used in low, moderate income countries. And there are even newer ones on the market, which are even going to be cheaper. But we can get these fetal ECG-derived measures, we can look at markers of brain function, and we can look at associations later. And very quickly, I can just show you that if you had a particular trajectory of autonomic development, which was what we consider the highest or best first, the highest trajectory, this is what your EEG power and the theta band looked at the day after you were born. This, if you had a moderate slope, you had a trajectory that didn't look to be quite what we thought to be normal, quite normal, you had lesser EEG power. And if you were on a trajectory where the slope was much more shallow, you had even less EEG power at birth. So again, heart rate variability during the fetal period, the trajectory of that, using RMSSD, that beat-to-beat variability, among other things, showed up. And again, whether you're in active sleep doing parietal theta power or this high-frequency beta power, something that we know had never been seen before and now we think can be exported elsewhere. And so this was based on our first phase 1, but now we're going to be looking at 700 of these babies to look at more meaningful, I think, interactions. But again, this is just to show that the autonomic measures also are going to be associated with, this happens to be gamma activity, high EEG power, based on your autonomic profile, whether you're in the lower third for the autonomic profile, the middle third, or the upper third, looking at the autonomic measures and how it is that, that your brain is operating at birth. Then how am I doing for time here, Jeff? 10-15 minutes. Oh, good. Yeah, in fact, my alarm just went off to remind me that that's what I had. Oh, that was good timing. Oh, okay. I'd like to talk about one last thing, which, again, I just thought I'd bring up a new one instead of the other cohorts, which is now another new emerging and I think interesting phenotype to be looking at, and it's the effects of maternal sleep position during pregnancy. Why do I care about that? Well, there's been recent reports, several in fact, that have been linking maternal sleep position to stillbirth risk and the mediating factor for infants, in this case for low birth weight. But there have been 4 more studies now showing women who slept on their right side more likely to have a late stillbirth, while on their left side they're half as likely. Another study out of Sydney suggesting supine sleep position is an additional risk for late pregnancy stillbirth, and one that just came out about a month ago saying supine sleep position the night before had a 2.3%, threefold increase rather, on increased risk for stillbirth. Well, why is that? Well, we've kind of known a little bit about this before, is that different-sided sleeping can affect a lot of physiological function, but in particular, what it can affect is the opportunity to occlude maternal vessels based on, in late pregnancy particularly, once this uterus and baby gets very heavy, you're much more likely to be able to have some occlusion or partial occlusion of all of these vessels if you are supine. We knew that as when moms and those of you in the room, when you went in for your ultrasounds that had those during pregnancy, you would have seen that they often asked you to turn a little bit to your side because if you were leaning back or you were supine, you tended to get hypotensive. There was this occlusion where your blood pressure would go up and your heart rate would have to go down to compromise that, to adjust to that. This portrayal is showing us, in fact, that in the left position, by the way, this isn't showing it so carefully, Because the vessels are asymmetric versus the uterus, if you're on your left side, there is no opportunity to occlude those vessels. So again, you may be hearing words like left is best, just like with babies back to sleep campaign. We may be coming out therefore in terms of reducing unexplained stillbirth, ultimately of a left is best for a sleeping position point of view. We measured here, though, we wanted to see exactly what happens. In the laboratory, we have this really nice non-invasive measure of stroke volume and cardiac output. You used to have a catheter to do these things. Now, by just having 2 electrodes that are placed in the right position, you can get measures of stroke volume and cardiac output and show, in fact, that sure enough, if you're in the left position, you have better stroke volumes and higher cardiac output than if you're in the right, than if you're in the supine position. These are in pregnant women during the last few months of pregnancy. And then we can do something, I think, which is even more extraordinary, and for my mind and our group here particularly, we have wonderful colleagues sitting here in the front that are coming to us from Milan, who are in the engineering school there, and are now rapidly going in and looking at these very complex signals to try to make sense out of what's going on during this time period, where we now can send mothers home with this monitor. Here's a fetal heart rate overnight recording, using that device I showed you a little while ago. I'm just happen to be showing heart rate. It gets even more complicated when you look at heart rate variability. But here's a fetus's heart rate when he or she is in quiet sleep. Here's this fetus heart rate in active sleep. Quiet, active, quiet, active, quiet. The purple is awake. I think immediately you can see the fetus is cycling in and out of sleep states, something we think can be an early marker of how that system is developing. We can go in and understand that we have to pay attention to these sleep states when we're looking at something like heart rate or heart rate variability, because again, as I mentioned before, if you don't know the state, you're not quite sure what number you ought to attach to that baby. And we can begin to look at changes overnight. And then we can start looking at specific sleep states like the quiet one. And then we can look at mom, which has been ignored. It's rare, in fact, that mom's heart rate will even be taken, or her pulse rate anymore when she comes in unless she's in a high-risk category, for what's going on during pregnancy, much less what's going on overnight. This kind of not very clean graph, but this yucky green color is when the mom is supine. This is what her heart rate is doing. Red is if she's in the right position, and left blue is when she's in the left position. You can see changes in her heart rate based on the different positions she's in. And so we have to think about what's going on with mom when we're trying to think about what's going on with the fetus. And on top of all of that, we have to pay attention to what our circadian rhythms we think that are emerging in heart rate. We kind of knew this before, that in most adults there is a circadian rhythm in heart rate, but we had no idea what was going on with pregnant women and how that might be affecting their fetuses overnight as they cycle in and out of different sleep states, cycle in and out of different body positions, and how much time they spend in each one of these sleep states is probably going to be the important metric. One last little monitor we can now just came out a little while ago, which we can add to this equation, because there's one other thing we should think about, which is not just fetal heart rate and maternal heart rate, but what's going on when moms are having contractions, whether they even be the Braxton Hicks ones, which aren't involved with labor, but clearly they're going to be affecting the fetus and probably overnight. Well, we can now get a measure of these, and this same monitor looks to be able to calculate fetal movements, different bouts of fetal movement all night long. It's kind of an interesting metric, as many of you know. Fetal movement, or the lack thereof, or the change in fetal movement, is taken as one of the earliest signs of distress. And so as you know, if you're pregnant and you notice a marked decline in movement, you get yourself off and go get looked at again. Well, now it goes back into the clinic. Nowadays, we're hoping with this onset of these many, many wearable devices, this information could be saved at home, stored, and sent to the appropriate healthcare people in some locale. We're going to be adding that to our armamentarium. So we see that fetal, infant, child, and maternal sleep can be seen this is where we're going right now as a marker of neurodevelopmental health. We can look at the effects of exposure. We can also look at the effects of different interventions. Another group that we're working with got Gates money to look at, they said, geez, you've had a marked effect on babies' growth by the new nutrition regimen, but have you looked at the brain? So we're going to look at that. There's another group that are doing a parent-infant training, and they're saying, gee, we're really seeing nice changes in parent-infant behavior, but could we go in and measure the infant brain as a function of that intervention? So that's in process as well. But we also see it as a mediator for all things important. If you want to destroy certain things, certain functions, for example, growth with children, it's do something and alter their sleep. Growth hormone hormone is only released during sleep. And so if you have altered sleep patterns and don't cycle in the appropriate way, you no doubt have a very good chance of altering growth, immune function. We all know what it's like if we don't get a good night's sleep for a few days in a row, how the vulnerability we feel that's been conferred for us to be able to catch what's ever floating out there. Attention and memory. If you really want to destroy your ability to attend and remember something, pull all-nighters if you're a college student and try to think about what you don't remember that you studied for that night. But even all day long, as you know, much of what's being remembered is being reactivated and consolidated during different sleep periods and emotional regulation. If you want to be a basket case, don't sleep for a couple of days in a row. Really, so much of our ability to regulate our emotions and our mood is based on, or at least is dependent upon, having good sleep. So next to the last slide, there's all this new hardware out here. Danny Bruner out at earlysignals.org. It's a wonderful group where they're trying to gather all this information. It becomes a serious big data problem and try to understand how do we put all the information we can gather from all these different devices. This is just the ones with babies. There are 10 times this. There are ones with mothers. There are ones for the rest of us as we're walking around in our daily lives where we're getting much more information about our health and well-being, and allows us to be able to use these kinds of tools and approaches and analytics to be able to look at many other prenatal exposures. We're very interested in, with Jay Gingrich, Myrna Weissman, and the group within the Sackler Institute, in the effects of SSRIs during pregnancy and depression with Catherine Monk. Affects what is early delivery. That is an exposure. You're now exposed to the ex utero environment much earlier than you ought to be. Gestational diabetes and obesity, fetal sleep abnormality, sleep position, placental function. We have a wonderful grant with people in London who are doing MRIs first of the placenta, again, to get a placental function, but looking at less expensive ways to look for the same markers once they figure out what's going on with the placenta with ultrasound and the other EKG monitors I mentioned. And then we're looking at all these other things here with groups in Colombia and other places. So I wanted to end here. I wanted to end by first thanking these people who were part of all of the work here and many other people in the audience that I didn't mention because we're engaged in just, this is like nirvana here for us to be able to interact with so many people who care about the outcomes of interest, but also understand there may be roots that may tell us a little bit about early vulnerabilities, early signs, and when we can go in and intervene. So I want to thank them, and I want to thank all our funding sources, and I want to thank you for staying awake. Thanks.